My project for the summer was identifying the role of macrophages in compensated cardiac hypertrophy. So just to give a little background, cardiac hypertrophy occurs when there is an abnormal thickening of the myocardium due to stress and pressure. And basically, the myocardium thickens because at the cellular level, cardiomyocytes grow in size. And the reason they grow in size is because cardiomyocytes are unable to regenerate. And it's only been recently proven that cardiomyocytes are able to generate in mice, but only during the first 7 days after they are born. And after that, their regenerative capacity is lowered greatly. So when we are talking about hypertrophy, there are mainly two stages. We have compensatory and decompensatory. During compensatory hypertrophy, this is very common when we exercise or when we're pregnant to deal with the increased stress. But after a prolonged period of time, the heart transitions to decompensatory hypertrophy and eventually heart failure. And recently it has been proven that the immune system can play a role in the transition from compensatory to decompensatory hypertrophy, and specifically macrophages. And when we talk about macrophages, throughout this presentation, I'll be referencing to mainly two broad classes: tissue residential macrophages and recruited macrophages. So tissue residential macrophages are responsible for maintaining homeostatic conditions and normal tissue development in the heart. And they are usually seated in the organ during embryonic development. But recruited macrophages, they are recruited to the site of stress and injury when the heart is inflamed. So during TEC, for example, or hypertrophy, we would see an influx of recruited macrophages. So another term I'll be coining throughout this entire presentation is angiogenesis, which is the formation of new blood vessels. Another word for it is microvascular density. So we know that microvascular density increases during compensatory hypertrophy and decreases when the heart is decompensated, but we don't know how. So our hypothesis was that cardiac residential macrophages stimulate an increase in microvascular density, reducing overall cardiomyocyte hypertrophy in a compensated heart model. So for our experiment, there were mainly two key aspects. One, in figure 1, as you will see, is we administered an antibody-based depletion method using the antibody CD115, which depletes all macrophages. And through flow cytometry and F480 and CD11B stain, we can see that we depleted all macrophages within by a factor of 2-fold, but we also found through flow cytometry that it preferentially depleted residential macrophages. So those are the ones in the box that you see there. And then in figure 2, we also perform transverse aortic constriction surgery, which mimics hypertrophy depending on the treatment duration. So in the heart, you can see that there is a tie between two arteries. On the left is the brachiocephalic, and then on the right, there's the left carotid. So from the point of the heart, right, it would be the left carotid and brachiocephalic. And the reason we tied it off between those two arteries is because if we tied it off after the brachiocephalic artery, that artery leads to the brain. So if we tied it off after that, that would constrict blood flow to the brain, resulting in brain ischemia and lower survival rates. And then our time point for our experiment was we injected the alpha CD115 antibody or the isotype as a control into the 8-week mice because that's when they usually reach adulthood. And then after a week, we would administer TEC. And then after a week, we would then harvest. So it was 1 week of TEC mimicking the early stages of hypertrophy. So as for our results, we first quantified cardiac hypertrophy and pulmonary congestion, which basically verifies TEC. So as you can see in the figure on the left, in figure 3, we quantify cardiac hypertrophy by taking the ratio between the weight of the heart and the body weight of the mouse. And we can see that there is a significant increase in the TEC group compared to the sham in both isotype and alpha CD115. But we did not detect a significant difference between the TEC isotype and the TEC CD115. And the reason we wanted to quantify pulmonary congestion as well is because when the heart is hypertrophic, fluids will leak into the alveoli since the vein that leads to the lungs is backed up. And as a result, that will lead to an increase in lungs. So if our TEC truly worked, we would also see an increase in lung weight, which we did here. We see a visual difference between the TEC and the sham in both groups. So in figure 4, I then quantify the cardiomyocyte cross-sectional area, And as a result, we do see a significant increase within the sham and tech in both groups, but none between the two. And how I quantify these images was I would take the images at 20x and I would try to get cardiomyocytes that were cross-sectional only, meaning that the cardiomyocytes looked as circular as possible. Because cardiomyocytes are usually the shape of a rectangular prism. And if we were to take pictures of non-cross-sectional cardiomyocytes as well, the endothelial cells that run through them, they would look like lines, and it would be much harder to quantify cells that are lines. So in the next image, you can see the benefit of taking cardiomyocytes cross-sectionally, because now the endothelial cells are all the green dots you see. So when we measured microvascular density, there were mainly two parameters, one based on the area of the entire image or the area that I am analyzing, and two based on the cardiomyocyte count. So when I measured microvascular density based on area, I didn't see a significant increase in the isotype tech compared to the sham, but I did see a significant increase between the alpha CD115 tech compared to the sham. But when I quantified it based on cardiomyocyte counts, I did find a significant difference between the sham and tech in both groups, as well as the alpha CD115 tech compared to the isotype tech. So the plus sign indicates that there's a significant decrease from the alpha CD115 tech. And although we did not see a significant difference between the alpha CD115 tech compared to the isotype tech in context of cardiomyocyte area, that is one area where our hypothesis wasn't completely correct. So next what I did was I measured macrophage abundance using CD163. So CD163 is a cell surface receptor found on a lot of macrophages, but more specifically residential macrophages. So in the green, you can see that I did the same thing as figure 4, in which I stained for cardiomyocytes, but I just stained them in the green channel at 488 nanometers. And then in the blue, you can see that it is DAPI, which stands for the nuclei. And then the red spots, those are the macrophages. So the reason I wanted to include the DAPI macrophages is because that's how I actually quantified them, because not all of the red spots are macrophages. And I could tell that they were macrophages because within the red spots, if I saw a little blue dot, that means I know it was a macrophage. So as for my results, I did find a significant increase in the isotype tech compared to the isotype sham. And then obviously in the alpha CD115, we found little to no macrophage levels, which is really interesting because in figure 1, doing our flow cytometry, we found that macrophage levels decreased by a factor of 2-fold. But when we did the CD163 stain, as you can see probably, it obviously decreased by more than a factor of 2-fold. So next, what I did was I stained CD163 and overlaid it with IB4, which will stain for the microvessels. And based on these images, I just wanted to include them here to show you guys that we do see macrophages in the isotype, but none in the alpha CD115, which further verifies the administration of the alpha CD115 antibody. So although we didn't see a reduction in cardiomyocytes or an increase in cardiomyocyte cross-sectional area, while I was taking those images, I did see an increase in collagen, which is basically fibrosis, in a lot of those images. And I wanted to quantify and see the difference between the two. And personally, this was the most interesting part of my project this summer, because I found a significant increase in the alpha CD115 tech compared to the isotype tech. And we verify all these significance using two-way ANOVA with correction, since we have two independent variables. We have the alpha CD115 administration and the tech surgery. So as you can see, the two shams in the isotype and alpha CD115 in figure 8 is very, very similar. But in the tech, there were two mice that were way above average in the isotype, which is okay, I guess, because it still led to a significant difference between the two tech groups. So as for the images, the ones stained in red were the myocardium and cardiomyocytes, and the ones stained in blue, or that like dark purplish hue, were the collagen. So all these images, I quantified them in image J. And yeah, so as for my conclusions, we noticed that macrophages contribute to an increase in angiogenesis, even though cardiomyocyte hypertrophy did not decrease. And based on microvascular density analysis, when we accounted for it based on area, it did not show a significant difference between the isotype and alpha CD115 tech. But when we accounted for cardiomyocyte count, we saw a drastic difference between the sham and tech in both groups, as well as a decrease in angiogenesis in the alpha CD115 tech compared to the isotype tech, suggesting that macrophages, and more specifically residential macrophages, are really important in orchestrating angiogenesis. But interestingly, we found a significant difference between fibrosis in the alpha CD115 tech compared to the isotype tech, which suggests that macrophages may possibly orchestrate the balance between angiogenesis and fibrotic tissue. Because since the heart has a limited regenerative capacity, if we can target genes that stimulate proliferation, at the same time, we suppress genes that produce macrophages, which hinder fibrotic response, we can actually potentially increase the regenerative capacity of the heart overall. So future studies should be aimed at differentiating the role of residential and recruited macrophages in cardiac fibrosis and angiogenesis. So this project was made possible by MSROP. I would like to thank Vicky and Heidi for sponsoring us. It's been a really tough summer, but all the writing seminars, all like Monday weekly meetings, even though they were really short, it really created like a sense of cohort. So I really appreciate it. I would also like to thank Dr. Joop van Berlo. I mean, like he's answered a ton of my questions. A lot of them were pretty stupid, I'm not gonna lie. So I'd like to thank him for being really lenient. And lastly, one of my postdocs from my other lab, Dr. Tim Keaton, I would like to thank him for coming today. I really appreciate it. So if anyone has any questions or critiques, please let me know.
Hi, Chen. Very nice work. I was just a little unclear. So the CD115 and 163, those are cell surface markers for all macrophages or recruited macrophages?
So CD163 is a cell surface receptor, mainly for residential macrophages. So macrophages that are embryonically seated in the heart at birth or before birth. And then alpha CD115, theoretically, it was supposed to deplete all macrophages, but through flow cytometry, we found that it preferentially depleted residential macrophages.
Mm-hmm. To about a twofold level.
Mm-hmm. Yeah. So yeah, the figure is a little bit tiny, but we can see here that macrophage levels, it's 40.9, and then it decreases to 19.9. So about twofold, yeah.
Mm-hmm. Mm-hmm. And presumably you wouldn't want to deplete them any farther, right? Or the host would be susceptible to all sorts of immune problems, right?
Yeah, that is true, yeah.
Okay, and I have another poorly informed question. What if you did the constriction before you injected CD115? So what if you essentially created the problem and then suppressed the macrophage response?
Well, after tech, macrophages would influx to the heart fairly quickly. So if we gave it a 1 week time point, so if we did tech first and then alpha CD115 first, my guess would be that we would already see an abundance of macrophages already in the heart. So I don't know if that would work as well, but yeah, good question.
Well, yeah, so you sort of have to do the antibody first. I guess I was just wondering, there was a week between antibody and tech?
Yeah.
So I'm wondering, well, like, what could you do to more naturally mimic an event, a cardiac event, without that long time span?
Because theoretically, if the CD115 antibody worked, it would also deplete recruited macrophages, which are blood monocytes. So if we didn't deplete those first, those blood monocytes would have already been recruited to the heart and differentiate into macrophages. So
Right.
that would kind of ruin the purpose of the experiment where we want to see the effects of macrophages in the heart. But if we injected it before, then we can see how it affects how macrophages play a role in tech. Does that kind of make sense?
Yep, it does. Yeah, thanks.
Thanks so much, Chen. Really well done.